Good afternoon, everyone, and welcome to today's investor webinar to discuss AdAlta's phase 1 clinical trial data and the development plan for AD-214. My name is Jane Lowe. I'm the Managing Director of IR Department, and it's a pleasure to support AdAlta on investor relations. So presenting today will be AdAlta's CEO, Dr. Tim Oldham, and the formal part of this session will be followed by a Q&A session. If you'd like to ask a question, you can do so at any time by entering your question into the Q&A function below. And with that, I'd like to now hand over to Tim. Thank you, Tim.
Thank you, Jane. Thank you, everybody, for joining us this afternoon. I can still see a few attendees coming in, so we'll let them have a few more moments to arrive. Very good. It's my great pleasure today to be able to announce further phase 1 clinical trial results for AD-214, and more importantly, our pathway forward to efficacy studies in patients. Right up front, I'd like to thank acknowledge the importance of our radiolabelled version of AD-214 that's been developed with support from the Biomedical Translational Bridge Program and MTP Connect, and thank particularly all our collaborators who've worked hard on this over the past year and a half to enable us to make the announcements and the plans we'll be sharing with you today. I will be making forward-looking statements today, and so the usual disclaimers apply. So the key message today is that AdAlta is electing to progress an inhaled version of AD-214 into efficacy studies for idiopathic pulmonary fibrosis. We've generated invaluable findings from both our clinical studies to date and also the radiolabelled AD-214 preclinical studies that have enabled us to map a pathway to conducting the next clinical trials in a much more preferred inhaled formulation rather than the intravenous route we've been using today. The key points I'll be making at various stages during the discussion today are firstly that the intravenous AD-214 has successfully completed the first phase 1 multi-dose cohort at 5 milligrams per kilogram and received ethics approval to progress to 10 milligrams per kilogram, maintaining the safety profile we observed in single-dose studies. Secondly, the preclinical development of radiolabelled AD-214 is complete and it informs the dosing and supports the early transition to direct lung delivery via an inhaled route of administration. The totality of the results we've observed to date provide a clear guidance to the future development of AD-214 via inhalation. This will be a much more targeted, convenient, and cost-effective route of administration than intravenous. Importantly, the timelines to initial efficacy studies in IPF patients are largely unchanged, and the imaging agent that we have developed will continue to inform dosing and distribution studies. The current phase 1 program will conclude having achieved its key objectives. We will not continue with the phase 1B program proposed in patients with imaging, and this releases cash and drug substance for the inhaled delivery bridging studies. So turning now to the results of our phase 1 multi-dose studies. A reminder of our phase 1 clinical program comprised two components. Firstly, a phase 1 protocol in healthy volunteers, which is underway. We reported the results of the single-dose studies in March this year, and we now report the results of the first cohort of the multiple ascending dose studies in healthy volunteers. We had planned a phase 1B protocol in IPF and ILD patients using our PET imaging agent. The preclinical development of that agent is now complete, but as you will hear later on, we're not progressing with this program. The phase 1 healthy volunteer results to date confirm the excellent safety profile we saw in single doses, now in multiple doses at 5 milligrams per kilogram. Reminder that AD-214 is an excellent safety profile as a molecule in single and multiple doses. We haven't seen dose-limiting toxicities or adverse events of clinical concern in the single-dose studies, and the only deviation from that we saw in the multi-dose study were moderate infusion-related reactions in 3 participants that received multiple 5 milligram per kilogram doses. 2 of these participants received active drug and 1 received placebo. Bearing in mind that that was 50% of the placebo patients and a third of the active drug patients. This means that these infusion-related reactions were most likely related to the formulation. They also rapidly resolved at the end of the infusion and were not associated with any concerning clinical laboratory results, and there were no adverse liver or other organ functions detected. The Ethics Committee and the Safety Management Committee have both approved progressing to 10 milligrams per kilogram, the next dose in the planned dose in this study. We continue to see evidence that AD-214 clearly engages the target receptor, CXCR4, in vivo. We see dose-dependent changes in the biomarkers of CXCR4. We see high and extended duration of receptor occupancy on the circulating T cells. And importantly, these biomarker responses were consistent across multiple doses at 5 milligrams per kilogram, showing no evidence of tolerance to the drug as multiple doses were administered. From a pharmacokinetic perspective, the pharmacokinetics continue to be dose proportionate. The peak concentration of AD-214 after intravenous administration and the total exposure in the blood increase in dose proportionate or more manners at up to 20 milligrams per kilogram. And importantly, again, they're consistent across multiple doses at 5 milligrams per kilogram. Again, there is no evidence of tolerance or drug-induced clearance of AD-214. And the rapid distribution we've seen from the plasma at all doses is consistent both with the rapid increase in receptor occupancy and saturation of receptor occupancy, but also with the preclinical imaging that we've observed. This slide shows those biomarker results at 5 milligrams per kilogram. We track 3 key biomarkers: the level of hematopoietic stem cells, or CD34 cells, as these are indicated here, white cells, or WCCs, and SDF1, which is the natural ligand that engages CXCR4 in vivo. Blocking CXCR4 would be expected to increase both levels of circulating CD34 cells and white cells, and also have a transient increase in SDF1. And this is what we saw in the single dose studies. What you can see here on the slide in the top right is that the time course of CD34 increases exactly matches in multiple dose studies and over multiple doses the profile seen in single doses. And in the plots below, you can see that for all 3 biomarkers, the levels of the biomarker at 5 milligrams per kilogram is consistent with what we observed in the single dose studies and is consistent across multiple doses. Again, as a reminder, confirming that we are seeing no evidence of drug-induced clearances, drug-induced tolerances that affect the pharmacokinetics. We've also continued to see high CXCR4 receptor occupancy. We don't have the data to show on this yet. It's still under analysis for the full profile, but at all 3 doses, we achieved 100% receptor occupancy for at least 24 hours, which again was consistent with what we saw in the single dose studies. In terms of pharmacokinetics, I mentioned that the maximum exposure and the total exposure were consistent. Again, it's important in a multi-dose study that these properties do not vary across multiple doses, so that our dose levels over time and with extended chronic use can be predictable. And that's what we've seen so far at 5 milligrams per kilogram. So on top of a continued good safety profile for AD-214 as a molecule, we've also now completed the preclinical development of the radiotracer. And the purpose of this work was to inform dosing and support routes of administration decisions. And this program has done this early, much earlier than expected for us. So this slide shows on the left the images of AD-214 on the left-hand side of the screen, and the middle image is of another Eyebody against a different target. And you can see that the AD-214 localizes very quickly or distributes very quickly to the liver of the mice, whereas the other Eyebody has a much more systemic distribution, which is what we would expect if there was no particular organ clearance. So what we conclude is that AD-214 substantially and rapidly distributes to the liver. This is consistent with what we'd seen in our rapid elimination from the blood. What we can also conclude, and this is important, is that this is unique, or at least it appears so far to be unique to AD-214. We've studied a couple of other Eyebodies so far in this same model, and none of them have showed this sort of distribution. So we think it's something unique to the AD-214 molecule, and more importantly, we think it's something unique to the AD-214 route of administration. The slide on the right, or the chart on the right, shows the detection of radiolabelled AD-214 on circulating white blood cells, or PBMCs. And this replicates again what we've seen with low doses of AD-214 in the work we've done with receptor occupancy. Importantly, when you increase the doses of AD-214, as we've done by adding 100 milligrams per kilogram or 20 milligrams per kilogram of unlabeled AD-214, we eliminate this signal, and that proves that we are actively engaging via the CXCR4 receptor. We're also able to see this effect in other organs where white blood cells accumulate or reside, such as the spleen and the bone marrow. So while we do have high distribution and localization to the liver, we're also able to demonstrate through imaging that we're engaging CXCR4 in mice and non-human primates. So what does that mean for our overall program? Well, as I mentioned, this really drives us towards the inhaled route of administration being a far more convenient and useful route than intravenous delivery. We know that this rapid liver distribution and clearance reduces bioavailability of AD-214. Essentially, we're having at least half of the drug is being dumped into the liver very, very quickly, and it's not available to achieve its therapeutic effect. We know and have continued to confirm that despite this, CXCR4 binds to AD-214 continues to bind to CXCR4. And it has done this in all of the preclinical studies we've done, including the efficacy studies, and this continues to support potential efficacy. In fact, we've achieved and demonstrated efficacy in the Blair mice and mouse model, for example, despite this high liver localization. And finally, the liver distribution does not appear to affect the safety profile of the molecule. We've found no localization in hepatocytes, which are the cells in the liver responsible for metabolic activity. And this is consistent with the lack of any observed changes in liver function or toxicity in any of our toxicology or our clinical studies to date. So we conclude from this that the direct lung delivery of AD-214 could achieve a therapeutic dose at a much lower levels of drug than intravenous delivery. We've also concluded that the radiolabelled AD-214 is going to continue to be an extremely useful development tool as we make the transition to inhaled delivery. So the totality of results then support this move to an inhalation route. The advantages of this are multiple. We've already talked about improved bioavailability, avoiding first-pass liver clearance and delivering AD-214 direct to fibrotic areas gives us greater flexibility around dosing schedule to optimize receptor coverage and should result in efficacy at lower levels of drug. The inhaled route of administration is going to be way more convenient for patients. They no longer need to come into clinic for intravenous infusions. It's less invasive and they'll have greater freedom of movement. The inhaled route offers enhanced cost effectiveness through the lower drug doses, meaning a much lower cost of goods, and significantly also lower healthcare costs for the actual administration. Patient self-administration is always going to cost the system less than IV administration in clinic. And finally, being able to develop route-specific formulations for AD-214 gives us the potential to partner AD-214 by indication with different routes of administration for each. And this broadens the number of potential partnering options we have for AD-214 in the long term. We will also be continuing to investigate the intravenous route of administration. We have some very strong hypotheses around what is driving the liver distribution. And if they can be solved, then that even further opens up other routes of administration and other indications for partnering. So inhalation is a perfectly feasible route of administration for AD-214 and idiopathic pulmonary fibrosis. Inhalation is already regularly used in IPF and other respiratory diseases. In fact, there are 4 inhaled IPF therapeutics in development today, as well as IPF patients routinely using salbutamol and steroids for symptom relief. We've already learnt that there is a significant amount of interest from patients in Australia where clinical trials are being conducted on an inhaled pirfenidone product. There are a substantial number of biologics already in development for inhalation. At least 2 are on the market. 19 others have reached clinical stages. Many of those are fragment antibodies or single domain antibodies. In fact, the majority of them have a very similar size to or span a size range that covers single Eyebodies at 15 kilodaltons all the way through to AD-214 at about 73 kilodaltons. And the majority of these are administered via solution for inhalation. And there are a number of off-the-shelf devices suitable for nebulization of liquid formulations that can be deployed without the need to develop novel devices. And these are specifically designed for biologics. So what does that mean for timelines and development plans overall? On this slide, you can see on the top panel the steps needed to undertake to transition to an inhaled formulation, leading to, in the solid coloured bars on the right-hand side, the next clinical trials in patients, which will be a phase 1b/2a program that will generate evidence of efficacy. We will need to do a small healthy volunteer bridging safety study prior to that. But these timelines, as you can see by comparing these with the timelines below for the current intravenous program, really get us back into the clinic around about the same time we would have achieved with the intravenous program in any case. And we would have needed to do some formulation optimization because of these infusion-related reactions, and we would have needed to do some extended toxicology in any case. But the rate-limiting step really is the availability of resupply of clinical grade AD-214. Earlier this month, we announced that we had secured resupply, and the timeline sets us up for having clinical material available towards the end of, or to the middle of 2023, the end of fiscal year 2023. And so you can see that we will be ready to start clinical programs when we are resupplied. Importantly, we're also going to see a number of interesting milestones along the way, and potentially much more along the way than we would have had under an intravenous route, demonstrating continued progress on efficacy and distribution using our imaging agent to confirm not only that we're distributing AD-214 to the lungs, potentially that we're distributing it preferentially to fibrotic lesions, and confirming that we're reducing the systemic distribution. And all of that should come through in the next 12 months. And then we'll have the usual milestones around confirmatory efficacy and toxicology ahead of starting our next clinical program. We do expect this may push out our partnering window for AD-214, and we believe that optimizing the value for ultimate commercial partners may happen now in FY23, once we have that proof that we are delivering in a preclinical model, successfully delivering the inhaled AD-214. A reminder of why we're doing this: IPF remains a significant unmet need, and AD-214 remains on track to be a first-in-class molecule addressing this $3 billion market, which is in dire need of improved treatment options. So finally, in terms of what happens next, we are concluding the current phase 1 program since it has now achieved its key objectives. And we will not proceed with the phase 1b program in patients with PET imaging. This releases cash, and just as importantly, it releases drug substance to enable us to progress the inhaled delivery bridging studies. In terms of near-term milestones, we've now pushed these out to the remainder of FY22. We have completed the preclinical development milestone, which we've announced today. We've announced the top-line results of the multi-dose studies in healthy volunteers today. We continue to expect additional indication preclinical data through the second half of this year. And we expect early in 2022 to be able to present initial efficacy data for inhaled AD-214 in an IPF animal model, and the first inhalation PET images to visualize the distribution of AD-214 in the lungs of healthy and fibrotic disease model animals. Our other milestones and our other programs remain on track and unaffected by any of these changes in respect of AD-214. So our pipeline update is as shown. Again, we continue to progress initiatives across all of our programs and are on track for meeting the milestones towards the end of 2021. An update on our vital statistics, if you like. We had 6 million in cash in the bank at the end of March. Our 4C will be due out shortly, and we have delivered a 43% return to shareholders over the last 12 months. So AdAlta continues to have a valuable Eyebody platform for multiple asset creation. It is designed for difficult targets. We know how difficult they are, and we continue to knock off those difficult targets. Our lead asset continues to have multiple indication potentials. We are targeting a $3 billion market initially, and we now have a very clear pathway to phase 2. Our collaboration with GE continues to progress well, addressing an imaging agent market worth $4 billion. And we continue to hold to our vision for growth. The team is done an amazing job to assemble and analyse the data that has come off in the last month from PET imaging and the clinical programs. And we look forward to delivering on the growth catalysts that I've outlined in the previous slides. And so with that, I will hand back to Jane to moderate the Q&A. Thank you for your attention.
Thank you, Tim, and thanks everybody that have already started submitting questions through the Q&A function. Just a reminder, if you'd like to do so, you can use the Q&A button in the bottom of your Zoom ribbon. So just the first one that's come through here, Tim. Thanks for the very detailed summary. Should we consider this as good news for AD-214?
We think it's good news. We've taken a lot away from our phase 1 results, which have clearly shown that AD-214 is a very safe molecule, and we've had enormously valuable results from the PET imaging. We're really pleased to be able to respond to those results very, very quickly. I have always believed that there had to be a more convenient way to deliver AD-214 to patients than an intravenous infusion. And the results we've presented today show that indeed the inhaled route should be a much more effective way to get the right quantities of AD-214 to those patients. Importantly, we've also now in a position where we can transition to that inhaled formulation on the same timelines at which we would have generated efficacy data with the intravenous infusion. So by and large, we believe this is really good news. We are on track to deliver our efficacy studies in phase 2 on the same timelines, but now with a much more convenient inhaled formulation that's patient ready. It's always good to introduce changes to a biologic as early as possible in development. It's much easier to do it before phase 2 than it is before phase 3. And so this is a real bonus to be able to make this change now.
Thank you. Just a question here on the platform. So what does this mean for the rest of the platform?
So we continue to believe that AD-214, that the Eyebodies are well set up to address difficult targets. We've studied a number of Eyebodies with our PET imaging agent. We've radiolabelled these as well, and that we do not see the same liver clearance and distribution with those Eyebodies that we have with AD-214. So from this, we conclude that it is very specific to AD-214. In fact, it may well be specific to the formulation of AD-214, but we don't know that just yet. And that means that for the rest of our programs, they're largely unaffected. We do not think this is a systemic feature of the Eyebody platform at all.
Thank you. And just another question on the platform. Can you expand on some of the other targets that are being evaluated?
So we haven't announced the other targets we're planning to put into development at this point. Obviously, we're working on and have announced the Granzyme B collaboration with GE Healthcare to develop a PET imaging agent for immuno-oncology. Obviously, we're working on CXCR4. The next 2 targets will be G protein coupled receptors. So these are a family of receptors that have of huge interest to the pharmaceutical industry and which have proven difficult for traditional antibodies to address. And we're working through in the late stages of finalising the selection of the next 2 of those that we put into discovery in our own labs.
Thank you. Okay, back to AD-214. If you choose the pulmonary route for delivery, will you retain the FC, which is the FC fragment, or will removal of this domain help improve delivery efficiency?
So that's a really good question. The first thing is that we do not currently intend to remove the FC. That would be changing the molecule. An important part of what we're doing today is delivering the same molecule that we've always been working on. So all of our manufacturing data still applies, all our manufacturing processes still apply. The safety data that we've generated in the phase 1 program to date is still relevant because there will be drug presumably that will transmit through the lungs into the systemic circulation. So we now know that that will be safe. And so we're far from starting from scratch again with the current molecule. We will look at, for purposes of studying inhaled delivery of other Eyebodies down the track, we may well look at the CXCR4 Eyebodies themselves, but our current plan is to maintain the current AD-214 molecule so that we can leverage everything that we've done without slowing down and restarting anything.
We've got a related question here, and I think in part you've answered it, but perhaps not specifically with relation to PK and PD. So how much of the clinical safety and PK/PD data can be translated across to inhaled, or do you basically have to start from scratch again?
So we know, no, we definitely do not have to start from scratch again. So our manufacturing process for the bulk drug substance remains unchanged. We expect and hope that we'll be able to make minimal changes to our IV formulation for use in a nebulizer. And so all of that formulation work and that stability work is going to be directly relevant and inform what we're doing. In terms of the safety data, we would expect that systemic safety is always going to be a concern. The fact that we've now demonstrated safety at systemic levels of AD-214 that are much higher than what we'd expect to achieve with an inhaled delivery means that that should not be a concern to ethics committees and regulators as we move to inhaled. We will need to do work around the specific safety profile via the inhaled route. That will be different to an IV route of administration, but that means that we're building on top of what we've already done very much so, rather than starting from scratch.
Thank you. Okay, so question here, why have you elected to finish the trial at this stage despite the fact you've got ethics approval to continue with it?
We've achieved everything that our phase 1 program needed to achieve. We've demonstrated the safety of the molecule in single doses. We've demonstrated the safety of the molecule in multiple doses, up to 5 milligrams per kilogram. And now we know that the right route of administration for IPF is through an inhaled route. That will deliver, we would expect, systemic circulation concentrations of AD-214 way lower than 5 milligrams per kilogram. So in that sense, we've got everything we can achieve out of the phase 1 trials. We also know that half the drug is ending up in the liver. That's not where we want it to be. And so there's no point continuing to study safety in phase 1 when we would need to make changes for intravenous in any case. We're not going to proceed with the phase 1b program because, again, that imaging has told us where the drug is going. It's told us in animal studies rather than having to do that in humans. And what we need to do now is utilize that same imaging tool to be able to demonstrate and inform that we're getting AD-214 to the right place in the lungs of patients in the preclinical animal studies we need to do as part of the bridge. So there was limited value to be obtained by extending the program in phase 1 or continuing the phase 1b program. My VP of clinical product development constantly reminds me that experimenting in humans is the most expensive way to do it. And so we can generate a lot more value a lot faster by saving the money from those phase 1 and phase 1b program, saving the drug product from that phase 1 and phase 1b program, and using that to progress our transition to inhaled.
Excellent, thank you. Okay, and a couple of questions here just on inhaled as a format. So first one is, why didn't you go straight down the route of an inhaled version of AD-214?
So hindsight's always is always a wonderful thing, but the reality is that the fastest way we could establish the safety of the molecule AD-214 was to proceed via IV administration. The other benefit of proceeding via IV administration was that it provided a safety data set that was available for all routes of administration and all indications. An inhaled route would have potentially locked us in purely to IPF and would not have given us the options to expand indications into other expand the indications of AD-214 into other organs. Clearly, we're not going to inhale AD-214 for kidney fibrosis, for example. And so there were significant advantages in progressing initially AD-214 in intravenous administration. We also thought that that would be the fastest route to generate efficacy data in a phase 2 program. Because the development of an intravenous formulation is much faster and a little bit simpler than an inhaled route. As it turns out now, given our resupply timelines, we're in the fortunate position of being able to make that transition to inhaled without slowing down our timelines for clinical efficacy. And by doing that, it's much more prudent to make that switch now rather than later on. So the intravenous was all about maximizing indication potential and maximizing or minimizing the time to efficacy data. We're not compromising that by making the switch now, and we're going to end up in efficacy trials and our next patient trials with a way more convenient formulation for IPF than we would have had before.
Excellent, thank you. So now a couple of extra questions just on opportunities around these different formats. So the first is, can you expand on the potential partnering opportunities using different routes of administration?
Yes, we can. So one of the challenges with commercialization of any drug is by indication is the fact that clinicians are actually pretty smart. And if you end up with different pricing for different indications, but the same format of the drug, clinicians will use the cheapest one. We've seen that with AMD, for example, where there are two very similar molecules, are essentially the same molecule, one price very high, one price very low. One is used in oncology indications and one is used in AMD. The retinal physicians very quickly worked out they could use the oncology one in AMD, and they do so. So you see this crosstalk between indications regularly. And what that means is that Big Pharma really prefer to partner the molecule as a whole, so they're not fighting over whose sale that was. Now, the way you get around that is by having very different formats and presentations for different indications. So if we've got an inhaled route that uses a nebulizer or inhaler of some kind, that presentation is going to be so different from, for example, the presentation we might need for a small intravitreal injection for AMD versus perhaps an intravenous injection for chronic kidney fibrosis. That would potentially enable us then to have separate partners for each of those indications. The benefit of that is that we've then got almost guaranteed that 3 indications are going to be developed versus a partner for the whole molecule may only choose to develop 1 or 2.
Very good, thank you. And so then now a direct question about a particular indication. So is there a case for developing this drug for liver fibrosis?
So that was obviously the first question we said is, have we got an amazing liver fibrosis drug or amazing drug for doing anything else in the liver? The short answer is probably not. We do know that the liver distribution is not driven by CXCR4, so it's not it's not wholly driven by CXCR4, so it's not an on-target distribution. It's more likely to be a method of clearance of the drug. We have some fairly strong hypotheses around what is happening here. We do know that the drug is not localizing to the hepatocytes, which are the cells in the liver that are responsible for metabolic activity. They're also the cells in the liver that go wrong in NASH, cancer, et cetera, et cetera. And so we're probably not delivering AD-214 in the way it needs to for a liver indication for fibrosis. So that's certainly not something that's an obvious outcome of this, but we have done the work to understand why that's not the case as well. But ultimately, it's a non-specific distribution of AD-214 to the liver, and that means that it's not ideal for a therapeutic use, and it's probably not going to the right cells either.
Thank you. Okay, just a reminder, if anybody would like to ask a question, we've got time for a couple more. Please do so using the Q&A function in the bottom of your screen. Tim, this next one's just on cash. So with cash released and savings from not progressing with the end of the current phase 1, how long is the current cash runway?
So we certainly have extended the runway. Clinical trials are really expensive, and so by not progressing with clinical trials, we've freed up a significant amount of cash. We are planning to spend that as rapidly as possible. I can't predict exactly how much longer the current runway is until we finalise the timelines with some of the contract research organisations that will be responsible for helping us with the bridging studies in animals and with formulation. But I can say that we do have an extended runway as a result of the money we've saved on clinicals. More importantly, I guess, is we've saved a significant amount of money on drug as well, both because we will have drug product available now to assist probably with the healthy volunteer safety bridging study for inhalation, but also with toxicology, and also because we're going to be using less of it. So the financial benefits happen over the longer term. But in the short term, we do have an extended runway. I'm not really in a position to be able to say how much longer at the moment, but it is longer than it was previously.
Okay, we do have a couple of extra questions coming through. Thank you, everyone. So one here, Tim, if IV AD-214 was effective in bleomycin mouse models, isn't it okay if it's distributed via the liver, i.e. we just now know where it goes?
So the good news is that that's probably the case. One of the obvious concerns would be that we're getting this amazing amount of drug ending up somewhere where it shouldn't. Is that a safety risk? And so far, we haven't seen any evidence of that. We've seen nothing in the toxicology studies in non-human primates. We've seen nothing in the clinical studies to date that suggests we're impacting liver function. We've also isolated we know it doesn't go to the hepatocytes in the liver. And so that is further evidence supportive of lack of long-term liver toxicity. But the reality is, with this much of the drug ending up in the liver, we know that we can significantly reduce the amount of drug needed if we can avoid this. And that takes significant pressure off our manufacturing improvement program. It significantly reduces the cost of goods, which gives our partners much more pricing flexibility to maximise market share than they would have otherwise. So it is never the right thing to have drug in a person that's been wasted because it's not going to the right place. So we need to make that move forward. Could we have continued with the intravenous formulation? Potentially, yes, we could have. But those challenges around the level of the total quantity of drug and the cost of goods at the end of the day would have been something that was not prudent to run with.
Great. I think there was another question here, actually, which perhaps you've just partially answered. So given the opportunities just discussed for other routes of administration, would safety and efficacy data from the remaining phase 1 MAD 10 and 15 milligrams per kilogram doses be useful under those scenarios and save time in the longer run?
Look, possibly. Given the infusion-related reactions we saw, we would be wanting to modify the formulation for AD-214 in any case for continued intravenous use. It's highly likely that standard antihistamines and hydrocortisone would manage these, and that's what the ethics committee approved the inclusion of for those higher doses. But if we did change the formulation to address these, then we would have needed to go back and do some of these studies again anyway. It would always be possible for us to restart this protocol. It's an approved protocol. There's no reason why we couldn't just restart it at some point down the track and do that relatively quickly if we needed to. So we don't really think there's a significant amount of value in continuing with that intravenous program right now.
Thank you. There's a technical question here. What particle size is problematic for smokers? And how does this compare with the formulation of AD-214?
So look, it's an obvious question about how do we get drug into the lungs of a patient with any kind of respiratory impairment. Now, it turns out that the particle size is important to drive the distribution of the drug into the right area of the lungs. So you have large particles, they will lodge in the upper airways. Smaller aerosol particles are needed to get to the distal areas of the lung where fibrosis is going to occur. This is well understood, and the technology exists to tune the particle size to ensure that the drug ends up distributed appropriately through the lungs. It's surprising how many patients with reduced respiratory capacity are actually able to and actively use nebulized drugs in particular, and even dry powder inhalers. So inherently, the fact that they have respiratory challenges is not something that we see as a barrier. And many of the nebulizers that we're looking at, for example, are actually breath activated, so they rely on being able to sense when the patient is actually breathing in order to release the drug. This is way different from the traditional asthma inhalers, where you're familiar with the patient having to actively suck on the puffer really hard in order to get the drug in. So these nebulizers don't require the same level of effort on the behalf of the patient in order to have drug administered.
Thank you. Okay, at this stage, this is our last question. So just a reminder to anybody that wants to jump in with a final question or two, now's your time. Use the Q&A function in the bottom of the screen. But this one, Tim, was the change to delivery route of administration guided by partnering discussions?
No, it wasn't. At the end of the day, we are a data-driven company. We're doing the right thing to deliver the right products to patients. And our focus has been on idiopathic pulmonary fibrosis. That's our first indication. That's where a lot of patients are actively following us. The right thing to do is to make sure that we could deliver a drug that was as convenient and as effective and as cost-effective as possible for them. And the data has driven us to this change.
Great. Okay. I did see one question just pop up in the chat, and we might make this our last one, Tim, just on the share price and what might drive positive momentum in the share price, which I'm sure you'll say we're working very hard to do, but I'll leave you with that, and then perhaps you might like to wrap us up.
Yes. Thanks, Jane. And thanks for that question. It's difficult to work out what actually drives the share price in this day and age. There's lots of factors going on, and some of them are inside our control and some of them are outside of our control. At the end of the day, our objective is to build value by adding multiple Eyebody-enabled assets to our pipeline. We continue to do that. The thing that we can control as the management team and the board is to continue to deliver assets into our pipeline and to continue to progress those assets. We believe we're continuing to do that. We believe that we are being transparent with the data that we're generating, so that the market can see exactly where we're up to. We believe we're sharing all of our strategies and strategies as they evolve. It's up to the market to tell us what they think of those, but we certainly are actively in conversation with our shareholders, potential investors and partners to ensure that we are absolutely listening to what the market is seeing and feeling. I think the benefit of the move to inhalation now is not only a more convenient formulation down the track for those patients, but it's also going to increase the amount of news flow that's going to happen over the next 12 months. We would have actually been pretty thin on the ground over the next 12 months, apart from the imaging from the phase 1B protocol, simply because we were then waiting for drug for the next program. So we actually now have much more rich data environment to come through that people will be able to see that progress. I think the imaging tool is going to be fantastic because it's going to provide a really visual and graphic perspective on how we're progressing and how our drug is behaving that we previously haven't had. And it's set to become a fairly essential part of our development program. But at the bottom line, we're a company with an amazing platform technology. Our Eyebodies are definitely capable of addressing difficult drug targets. We have a strategy to add more products to our pipeline. We have 2 in the pipeline today. We've provided an update on AD-214 now towards a more patient-convenient inhaled formulation. Our GE collaboration progressed to preclinical development last quarter, and we're on track to have 3 more programs in the pipeline by the end of the year.
Excellent. Well, with that, Tim, shall we thank everybody for their time and look forward to keeping you all up to date as AdAlta continues to report on those milestones. Thank you, everyone.
Thanks, Jane. Thank you, everybody, for joining us today.